Hello, my name is Ayoob. I'm Redi, and my research is on the topic: does pasteurization destroy vitamin C? First of all, before we even begin discussing the issue, we must know the definition of vitamins, and they are organic compounds which our bodies need but does not produce. Vitamin C, also known as ascorbic acid, is a vitamin essential in the making of collagen, which is the protein that strengthens bones, tissue, teeth, etc. Vitamin C has various other health benefits such as protection against cancer, heart disease, and may even improve one's sex life. A lack of vitamin C may cause fatigue, depression, and in some cases, but not a lot of cases, scurvy. Foods that are rich in vitamin C are mainly vegetables and citrus fruits, especially oranges. As you can see, they are great sources of vitamin C. Pasteurization, on the other hand, is a process developed by Louis Pasteur which involves heating food to usually 100 degrees Celsius, which kills bacteria and extends the shelf life of the food. It's mainly used in the dairy industry. Coming to my hypothesis, as you can see, our bodies need vitamin C, and pasteurization extends the shelf life of foods. However, heating vitamin C may destroy it, so my prediction is, if orange juice is pasteurized, then it will lead to decreased levels of vitamin C concentration. So coming to the experimental design, the way we did this experiment was 10 oranges were cut in half with a knife and juiced using an orange juicer to get around 50 milliliters of orange juice from each half of the orange. Next, the orange juice was strained using cheesecloth to eliminate pulp. After that, around 10 beakers of approximately 50 milliliters of orange extract were obtained. Then, the juice was poured one at a time into centrifuge tubes. 10 labeled pasteurized and the other 10 labeled unpasteurized. A centrifuge was prepared and each tube was pelleted for 5 minutes at maximum speed. After that, tubes labeled pasteurized were placed in preheated water of 80 degrees Celsius for at least 10 minutes. The pasteurized orange extract was then returned to ice and was left to cool down for 10 minutes. After that, 1 milliliter of pasteurized orange juice extract was mixed with half a milliliter of DCIP and put in a spectrophotometer tube. The transmittance is set to 100% and absorbance at 520 nanometers was measured for the cuvettes that contained the pasteurized extract. The same process is done with the unpasteurized extracts. At the end, we have data for 10 unpasteurized samples and 10 pasteurized samples, which is then recorded on Appendix 3, page 3. As you can see, using the standards given to us, a graph was made, a scatterplot graph, and omitting the various irrelevant data points at either side of the plateaus, we could then get a standard curve which translates into an equation which we can then use to figure out the vitamin C concentration. Coming to my results, I got 10 pasteurized samples and 10 unpasteurized samples, with the pasteurized average being around 42.6 and the unpasteurized average being around 42.4, each having a standard deviation of around 6 and the p-test value being 0.94. Coming to the bar graph accurately shows how similar both pasteurized and unpasteurized concentrations are. As you can see right here, the standard deviation of both are the same and the values are basically the same, which disproves my hypothesis. And also, since my p-test value was above 0.05, my null hypothesis is supported, which also disproves my alternate hypothesis, which is that pasteurization affects vitamin C concentration. Discussion. I was surprised that the data contradicted my hypothesis, since I was sure that the heating process in the pasteurization would denature and destroy the vitamin C in the orange juice. The data showed consistently that it was the other way around, and one thing I may change about my experiment is that I might pasteurize the orange extract at a higher temperature, just to make sure that there was enough heat being supplied for the experiment. The implications that this might result in the real world is that it doesn't matter if you pasteurize or don't pasteurize orange juice or anything related to vitamin C, because at the end of the day it doesn't change or decrease the amount of vitamin C it has. So if you want to pasteurize orange juice to increase its shelf life, then go ahead. Conclusion. According to the p-value and the graph, there is no evidence to suggest that pasteurization leads to decreased level of vitamin C concentration. In conclusion, the hypothesis that I had that pasteurization of orange juice led to decreased vitamin C has been disproven, and pasteurization, according to the data, does not affect vitamin C at all. For references, I have Appendix 3, Appendix 4, and Appendix 6 by Wimmers, Tsuji, and Deniston from Department of Biological Sciences at Towson University. Thanks for listening.